Hello, everyone. Very welcome to this talk within the campus party. I'm Guido Sanguinetti. I'm a professor at SISA in Trieste, as well as a professor in the School of Informatics at the University of Edinburgh, where I've been working for over 10 years now, in the field of machine learning and its applications to what was once called systems biology and biomedicine in general. And what I would like to do today is to give you a brief introduction to some of the major questions and the ways we are trying to address these questions in applying machine learning to biomedicine. So the title is really trying to capture the fact that we go from thinking about molecules within individual cells to patients and trying to connect these levels where understanding the basic biology of disease can then inform us to target therapies better to individual patients and try to achieve better results in terms of prognosis, of diagnosis, and eventually, of course, of therapy. So I would like to start with maybe a slightly provocative set of questions about what is really my attitude towards machine learning, AI, and big data in general. And this has been matured over the course of quite a few years. So hopefully that can give you some insights as to why we do the kind of machine learning that we do. And I'll also introduce a little bit more technically what kind of machine learning we do and why it can be applied to contexts as different as individual patients and individual cells. And I'll finish up with a couple of examples of the type of work that we do in both the analysis of single cell data and cancer data. I appreciate that some of these things will be of an introductory level, some others will be a little bit more advanced, but I'm very happy if you just get a gist of what we're trying to do and why it is important, that would be great. And if some of you feel motivated to look further into these things, please try to find my papers online or get back to me, whatever. I'd be very happy to hear anything. So, when I was starting to prepare for this talk, I thought back to a conversation I had almost 10 years ago with a fairly famous statistician that in those days were the early days of big data. And everyone was talking about big data. We have lots and lots of data, and this will be transformative. And this guy asked a rather pointed question of, what the hell is the point of having 1 billion independent and identically distributed data points? And what did he mean by that? Well, generally, what we want to extract from data are summaries, statistics. So we could sample the population and try to figure out what's the average height of Italian males, for example. And we could take, 20 students from Milan University, or we could take 200 people from Milan, or we could take 20,000 people from Milan, or 2 million people from Milan. And what we will get is that we will get increasingly more precise estimates of the same number. So his point was, well, do we need big data? We can probably get the same estimates from slightly smaller data and just now sample the data. Now, the question, of course, wasn't entirely naive. We now know very well that, in fact, the power of big data is in identifying structure in high-dimensional data. And for those of you that have been following a little bit the developments of AI, you'll be perfectly aware that things like neural networks have a somewhat magical property of managing to avoid what is called the curse of dimensionality. So we have very high dimensional data, lots of data, but still wouldn't be enough for a classical type of analysis. But yet some techniques that have been developed in the wake of big data enable you to essentially find low dimensional structures in this high dimensional data, which are extremely useful for predictions. Nevertheless, the point of my colleague's question of what's the point of having 1 billion independent and identically distributed data points was slightly different. And his point was that we very seldom have data that is exactly the same in the way it is generated. So what does that mean? Well, for example, we might have huge amounts of data, of text data, but it is text by different authors that write in a slightly different style. So it's not IID. The way the distribution of words in the way I write is very different from the distribution of words in the way someone else writes. For example, someone that writes technical specifications of bicycles is very different language from the one used by a scientist. But more pertinently, we could be interested to look at analyzing medical data coming from scans. And different hospitals may have different machines for the scans that may be of different age or different makes. They will have slightly different settings. So the data is not IID. It's slightly off between one and the other. Obviously, they're related, but they're not identically distributed. Or we might have some biomarkers from different patients, and obviously different patients are different individuals. We can't assume that they would respond to therapy in exactly the same way. So all of these are examples where there is a degree of individuality to the data points. The data point may be a piece of text, or maybe a patient, an actual individual. And sometimes it is important to make predictions that are tailored to the data point, to the individual. Another way of thinking about that is to do an ethical experiment, okay? So we are now starting to be in an age where we interact with computers in a completely different way from the way we were interacting with computers 10 or 20 years ago. So now imagine you are in this scenario, computer says, well, computer might be saying, okay, buongiorno is the same as good morning, is the same as guten Morgen, because we are trying to have our computer work on a machine translation scenario. Yeah, that's perfectly fine. This kind of observation from the computer is helpful. The computer may say, okay, I'm analyzing this picture and it contains two cats, a table and a car, and I'm perfectly happy about that, partly because I can verify this information myself readily, and partly because, it's not a life or death type of information. But maybe the computer one day will tell me, based on your genetics and your lifestyle, I estimate that your remaining life is 27 years, 3 months, and 8 days. Now, is this an acceptable thing for a computer to say to a person? Well, everyone has his or her own opinion, but I personally wouldn't like to hear that because, A, I don't know why. How did it get to this conclusion? And if I don't know how the computer reached this conclusion, I can't know what I can do in order to change it. And finally, I have absolutely no way of independently verifying it. It's obviously a very important matter, and there is no way I can find out whether it's true except for waiting the 27 years, 3 months, and 8 days and see whether I actually die on that day. So I think this illustrates that really at a fundamental ethical level, we have to distinguish between purely predictive techniques applied in scenarios where the individuality is not so important, completely different from scenarios where you're making a prediction about one individual that is of extreme importance. So I would propose to think about 3 axes along which we can classify the acceptability of our interaction with AI. One of them is the importance of the predictions for the person that is seeking the prediction, the ability to act on the predictions, and the ability to independently validate the predictions. So if it is a prediction of relatively low importance, like are there cats in this picture, and I can easily find out myself whether the computer is right, well, then I don't need to find out how it got there. If it gets the right answer enough times, then I'm happy. But if it is something of extreme importance that I need to be able to act on, and I can't validate it, such as a prognosis or a diagnosis of a disease, I need to understand how the computer got there. And this is the whole foundations of why people bang on about explainable AI and AI that interacts with humans in a way that is transparent. And that's why it's a huge problem, because what we have now is that we have some methods that on tasks such as computer vision or text analysis, machine translation, we have methods like convolutional neural networks or transformers that achieve fantastic performance, but we don't understand why. And if we try to just apply that to predictions that have a real importance to the individuals, we might be in trouble. Not only, just to make another point, we can't, in the case of predictions where there is real importance attached, we always need to be able to associate a level of uncertainty. So the computer might state that something is certain, but nothing is certain in the future. Anything that is a prediction should be attached with an uncertainty quantification. If I have an important prediction that I need to be able to act upon, I need to be able to know how reliable is this prediction. Is it something that will happen with 57% probability or with 99% probability? And I need to be able, when I look at the way the conclusion has been reached, to understand how reliable each step of the prediction is. I need to be able to find out which things the computer is certain about, which things it's not entirely certain about, and therefore I might need to gather more data. So, having said that, this is kind of the prologue as to why, when we're trying to apply machine learning to biology and to biomedicine, we need to think about a slightly different machine learning from the one that we apply to computer vision or to machine translation. Now, what type of machine learning? Well, what is machine learning? Everyone is talking about machine learning these days. And I would venture that most people that talk about machine learning don't really know what they're talking about. So I went to the source of all knowledge, Wikipedia. And Wikipedia gave me a fairly opaque definition of machine learning as a subfield of computer science, which I agree with, which gives computers the ability to learn without being explicitly programmed. Now, this is a very vague. So what do they learn from? What does it mean without being programmed? And what does it mean explicitly programmed? Well, the reality is that machine learning is a family of algorithms. Okay? And what these algorithms do, they start with data, which is a key thing that is missing from the Wikipedia definition. Machine learning needs data, and mathematically encodes patterns within these data that then can be used to make predictions. For example, in a supervised learning task, I might have lots of images with labels saying contains cat, and try to find out what is the pattern in pixels that associates with cats, so that when I see a new set of pixels, a new image without a label, I can make a prediction. And there's a huge variety of machine learning algorithms that encode predictive patterns. In some cases, you may want to hardwire the pattern because you have very high levels of certainty about how something works. So we have machine learning algorithms that are very restricted in their flexibility, but very well tailored to particular problems. And sometimes these are important problems. On the other hand, we have machine learning algorithms that make very, very few assumptions and require large amounts of data to find patterns. So machine learning is a broad church. It's not just neural networks, it contains a lot. And in practice, what it tries to do is best understood in terms of random variables. Okay. Now, here is a little bit more technical, this part of the talk, but I know it's not as interactive as it would be in person. But if you have any questions, just let me know somehow, and towards the end of the talk, we'll try to address them. But generally, the patterns need to be relationships between different variables. Okay, the variables could be the pixels in an image, the intensity levels of the red, green, and blue in an image at single pixel level, and some other variables may be the category of image that something is. So we have a number of random variables, which are generally divided into two groups. Let's call them inputs and outputs, X and Y. And what we want to do is to learn from data how these random variables are distributed. Very often, we have some additional variables that we cannot measure directly. For example, we might be able to measure the age or some biomarkers for a patient, but we cannot measure the stage of the disease directly. We have to infer it from data. And what we want to do is to leverage this joint probability distribution to infer a posterior distribution of unseen variables given observations. And finally, we want to use these probabilities that we've learned to predict what new data will look like. How do we do that? Well, the framework that I would advocate, which is the more general framework, and every other type of machine learning is somewhat a specification of this framework, is the framework of empirical Bayesian inference. So we formulate our models in terms of random variables. We place prior distributions about the way the random variables interact. We have a set of free parameters that give us the flexibility to learn from the data. And we learn these parameters by maximizing the evidence of the data given the model. And then we do posterior inference using Bayes' theorem. Now, this is terribly hard computationally. It's a big computational problem. It's probably the biggest computational problem in computational statistics, and to some extent also in machine learning. How do we compute these distributions given data is highly non-trivial. And a large amount of machine learning and statistic research goes into how we solve this problem approximately and efficiently. Now, this talk is not meant to be a technical talk, and this is probably all the content, the technical content. What I want to show you now is how we can adapt this kind of way of thinking to deal with some real problems. So what would be the beauty of this approach is that you can formulate the models in terms of relevant observed and unobserved questions, and you will come up with models that will represent graphically like this. This is something that is called the graphical model. We have a set of variables. The shaded ones are the observed variables. So we see here we have an R variable that is observed, an X variable that is observed, and we have other variables that we think are important that we cannot observe, but we have a good idea of how they interact with each other. And what we want to do is to use the Bayesian framework to learn from sets of observations of X and R what is the distribution of these unobserved variables. And how, if I then find out, for example, I'm given only X for a next data point, what would be my prediction of R? Okay, so this is the general framework of Bayesian machine learning. It builds models formulating every relevant variable for the problem as a random variable and postulating some relationships between the random variables. These can be very broad, so we can have very flexible models that can adapt, can be nonlinear, whatever, but they are still by design interpretable because I know exactly what one of these arrows means. It's a conditional dependence representation, which I can then try to understand and probe empirically by doing other experiments or looking at more data. So this is the general high-level framework where we're going to work. The kind of problems that we're interested in are the problems where we are trying to understand how the basic components of cells work together to produce life as we know it, and also what goes wrong when something doesn't work the way it should. For example, some of you may have heard quite a lot of talking about something called precision medicine or personalized medicine, which are broadly related systems. Now, we have a huge problem that human beings are not all the same. That's kind of a great thing because it makes the world varied and interesting. But it's a problem from the point of view of medicine because we frequently, with many diseases, observe that one individual responds well to a therapy, another individual with seemingly the same disease and maybe not even very dissimilar life history does not respond well. This is particularly problematic for cancer, but it is a common problem for many, many diseases. And this has led to the increasing realization that, since humans are not all the same, treatments cannot be all the same for the same human. How do we understand how to target treatments for particular humans? Well, statistically, in biostatistics, people call this problem stratify. So you have a population of patients, and they're not all the same. But of course, we cannot develop a specific drug bespoke for each patient different. What we need to be able to do is to understand what is the structure within the population of patients and what makes the difference between these structures, so that then we can target particular treatments to particular groups and hopefully achieve the best results on everyone. All of this precision medicine essentially has started gaining traction when sequencing of genomes has become really cheap. So as you probably know, it took more than $1 billion and several years of a huge multinational effort to sequence the first human genome around 20 years ago. It is now possible to sequence one human genome for a few $100s in a few hours. So technology has moved super fast, in fact, even faster than computer technology, which means we are now in a position where in many countries, if you are diagnosed with cancer, you're going to have your genome sequenced routinely. Why? Well, because we want to understand what type of cancer, but also we want to understand what type of person we're dealing with. So the context of the genetics of a person may be important to determine a treatment. Not only the genetics, of course, lifestyle choices, those were accessible much earlier. And for a long time, doctors have been prescribing different therapies based on the patient. However, we are aiming to arrive to a situation where each patient has a huge amount of data attached, the whole genomic sequence, plus potentially the whole history of their, for example, health records and so on. We would like to be able to use all of this personalized information, feed it into a machine learning algorithm, and obtain a personalized prediction which is traceable. We can tell which bit of the history or of the genetics informs each decision suggested by the algorithm. So here we have a scenario where we have huge amounts of data, but it's all huge amounts of small data. Each patient has a modest amount of data about the patient, but taken together, we have lots and lots of data. And we need to use machine learning not to make average predictions, but to make predictions that are relevant to each patient and tailored to each patient. Another topic where I'm particularly interested in is the analysis of molecular data coming from individual cells. As I already mentioned, one of the big drivers of the difference that we see between different patients is that the genetics is different. But there are further levels of variability because we are actually starting to realize that each cell is different. It's not different, each cell in my body is different, not in terms of its genetic content, most cells in my body are almost exactly the same in terms of the DNA, but they're different in terms of how genes in different cells work. And part of that explains why my muscle cells are different from my liver cell or from my heart cells. But part of this variation is intrinsic. So two different muscle cells will be slightly different. So we want to understand, out of these differences between individual cells, which differences come from the cell type, which difference come from the individual cell, and if we are doing a medical study where we have taken single cells from different patients, which differences come from different patients, and which of these differences are explained by the different genetics of the different patients, which differences are explained by the different lifestyle of the different patients, which differences just seem to be intrinsic. So at the moment, and really in the last 10 years, we've had fantastic new technologies that have enabled us to measure the expression of all genes in individual cells. That's what you're seeing here. Single-cell RNA sequencing measures the expression of every single gene into every cell in a group of maybe a large sample, could be 10,000 different cells. We can also measure other genomic features, for example, DNA methylation or chromatin accessibility, whatever. But all of these are characterized by having many cells measured all at the same time, of the order of thousands. Each of these cells has got its own high-dimensional feature space, the expression of tens of thousands of genes. And we want to be able to differentiate what makes the difference between different cells, what makes the difference between groups of cells. What does a cell of a certain group look like? If I were to see a new cell of that group, what would be its expression, its prototypical expression be? Why do I want to do that? Because then I may be able to compare the cells from different patients, or the cells from a healthy tissue from the same patient and the diseased tissue from the same patient, and understand what is changing at the molecular level. So again, we are in a situation where our data is huge, but is made up of lots of bits that are not big. Each of them is an individual cell and is a single data point in high dimension. So even though these scenarios are very different, we're talking about humans, whole trillions of cells, organism, and individual cells, we have very similar questions. We want to understand what are the important differences between the different cells, or what are the important differences between the different patients. Are there stratifications, are there groupings, are there structures in the population that are relevant for the disease progression? Is there a developmental process going on? So we may have many patients, all with the same disease, but at different stages. And that's not a variable that we can measure typically. We have some proxies. Doctors have been taking measurements and associated them with the stage of the disease, but that's an approximation. There is a conceptual, continuous change on the disease, and can we access from data that type of variable? So the general technical problem that I'm trying to introduce you to is what is called transfer learning. So we're not in a situation where we have millions of independent and identically distributed data points, and we are trying to find some very nonlinear associations between pixels and an output variable. We're in a situation where we have many, many thousands of data points, but we want to be able to predict for one data point. We want to learn what we can share in terms of information between the different patients while retaining individuality in the patients. So I'm going to give you two examples, one from single-cell biology and one from medicine. And then I'll wrap up and you'll be able to ask me some questions. So the first example is exactly the scenario where we can measure something about each individual cell. So this is an image from a paper that appeared in Science a couple of years ago, from the Ecker lab in the US, where they took cortex neurons from a mouse, and they measured what is called the epigenetic information in each cell individually. So epigenetic information is believed to be something that determines the behavior of the cell. What is this epigenetic information? In reality, it's a huge, very long string of zeros and ones, which is the DNA methylation status of every cytosine followed by a guanine. They did this for several thousand cells, and they identified several different groups of neurons within the cortex. Now, this is interesting because, of course, we have a very rudimentary understanding of how brains work from the molecular point of view. We don't understand. We know that brain needs to have many, many different types of cells interacting in order to produce high-level behavior, so cognition, for example. But we don't understand what are the relevant groups from the molecular point of view. And this was a first attempt to understand what are different neurons within the cortex based on their epigenetic status. This is done by a technique that is called single-cell bisulfite sequencing. But the problem, so I said, it tries to measure a very long string of several million zeros and ones, but it can't measure all of them. Only measures in one cell maybe 5% of all the possible zeros and ones. And once we have that, we have essentially each cell is associated with a very high dimensional feature space, which is only very, very sparsely as observed. So we need techniques. If we want to tease apart different types of cells, we need techniques that can handle these very large amounts of missing data. So generally, we can't use the data at face level in order to be able to understand, for example, what is the methylation profile of individual genes in every cell. There is not enough coverage, as it's called. And on top of that, not all the cells are equal. We suspect that there are different groups of cells. So the idea here was to come up with a machine learning model that would enable us to essentially identify groups of cells and simultaneously in-paint the different CPGs that are missing. So we want to use this limited data, but the fact that we have lots and lots of limited data to find out what are the groupings and what should be the prototypical profiles. And this was a paper that we published last year with my former student, Andreas Capurani, who's now a research fellow in Edinburgh. And the idea, again, is to combine individual cell level likelihood with a prior that couples the different cells. And this prior encodes the fact that there should be groups of cells that are epigenetically very similar. And if we do that, that's a graphical model. So as you see, we have a fairly complicated mathematical description in terms of probability distributions. But all that is saying is that there are k clusters, and each cell in the cluster should be broadly the same from the point of view of the methylation status, which is what we observe. And we showed that using this approach, we were able to predict very accurately the missing methylation bits that we were trying to in-paint in this long string. And we were able to identify different groups of genes, which were radically different in some different groups of cells, which were sometimes radically different in terms of their epigenetic state. So for example, we had 3 clusters, and that this particular gene, which is a relevant gene for development, we could see that one group had very, very low methylation, while another one was characterized by low methylation at the transcription start site and high shoulders, both in the gene body and upstream. So this is an example that enables potentially the biologists to tell, hey, there are groups here, and what is driving the different groups are these particular genes that look very different in the ways their epigenetics is. And then, of course, these kind of efforts, we work on the methods development, but we work in close collaboration with biologists that then can take this information further to develop more their understanding of how, in this case, the embryonic stem cell development was working. The other application I wanted to briefly mention in the last 5 to 10 minutes is an application of these ideas to cancer heterogeneity. Now, all of you are certainly aware that cancer is a huge burden on the healthcare of particularly advanced nations, and it's been so for a very long time. So in the 1970s, so about 50 years ago, Richard Nixon, the President of the United States, declared war on cancer and said pretty much the same way as we've put man on the moon in a few years, we are going to eliminate cancer in a few years. And now 50 years later, I'm here talking to you about cancer heterogeneity, and we are in a situation where the burden of the disease on the health services is as large as ever. And some cancers have seen dramatic improvements. Breast cancer, some types of breast cancers are curable now. But many cancers are still one of the major sources of mortality and morbidity across the world. And why is that? How come we were able to put a man on the moon, but not to cure cancer? There are many reasons. But one of the main reasons, in the way we understand it now, is that there is a huge amount of heterogeneity in cancer. So different cancers, even the same type of cancer, say the same prostate cancer, in different people are different. Not only within the same person, a cancer at a relatively advanced stage may be within itself different. What does it mean? Here you have a picture, idealized picture, where we have a tumor, and you can see there are different types of cells within the tumor. Why? Because cancer works by the acquisition of random mutations. They appear at a much higher rate than they would appear in normal cells. And some of these mutations are advantageous, and this leads essentially to a micro ecosystem within the tumor, where there are several types of tumors coexisting. That's a massive problem, because we can target a therapy to one, for example, to the pink cells and kill all the pink tumor cells, but this may not affect the yellow cells. So the tumor will shrink, might even shrink to a size that is no longer detectable, and doctors think that the patient is cured, and a few years down the line the cancer will come back, and that normally is terrible bad news. This is due to within cancer heterogeneity, as well as a huge amount of interpatient variation. And this arises, as I was saying, because there is essentially a little ecosystem within the cancer. So every time a cell divides, new mutations are acquired, and some of these mutations give an advantage from the point of view of evolution to the carriers, creating what is called the clonal expansion. So that cell starts reproducing faster. And then maybe some of the progeny starts reproducing faster. And in an idealized scenario, you would see a phylogenetic tree for each cancer. And each of these edges means new mutations that have arisen. And some of them have given some selective advantage, meaning that have given rise to different clones coexisting. Now, this is potentially life-saving information. So if we were able, given data from a cancer, to reconstruct its evolution and find out where in this trajectory we are, then we may be able to target therapies in a way as to avoid the relapses that we so frequently see. Now, how can we do that? Well, in principle, going back to the picture previously, we could, instead of taking, when we take a piece of tumor, we could take multiple spatially segregated bits, which will contain different mixtures of the different types of cancer cells. And from that data, we could try to reconstruct a phylogenetic tree pretty much in the same way as we reconstruct phylogenetic trees from existing, say, bacterial species. And we find out how one is related to the other and which mutations determined when the tree splits. Now the problem, to get to a tree of this level of detail, we would need to have many, many, many samples. Now we can't get from an individual, from a single patient, which has a single tumor, so many independent samples spatially segregated, because hopefully we catch the patients when the tumor is not too big. And technically, there is a limit to what we can extract. So what was happening until a couple of years ago was that people were trying to reconstruct these trees from each patient using 4 or 5 data points. So for each patient, we would take the tumor out, and we would sample the tumor at 4 locations and try to reconstruct a phylogenetic tree. And what would happen in that case is that we would get very, very low levels of information, because of course, on top of the evolutionary process, there is a huge amount of randomness. And that means that essentially it looked as if every tumor was completely different. What we did with a couple of colleagues from London, and Giulio, who's now just started an independent researcher position in Trieste, not at CISA, but at the university, was to try to do the transfer learning trick. So this is this TL here. So think that each patient was a different trajectory, but from the same evolutionary process. And so instead of learning different phylogenetic trees, one per person, we inserted a constraint that the different phylogenetic trees should be as similar as possible. And that allowed us to basically find macro groups, a stratification of the patients based on what was their evolutionary history. So here we would see, for example, that patient 1 and patient 3 have very similar evolutions, while patient 2, 4, and 5 have a bit different but similar between the 3 of them. Is this relevant? Well, conceptually, it's very interesting. Yeah, it's a new way of stratifying based on a feature that is not at all evident. You know, we're not stratifying based on one biomarker or whatever. We are stratifying based on the evolution of the whole tumor. And in practice, what we could show is that if you evaluate then on a larger data set the probability of survival on cohorts that were associated with different types of tumors according to our stratification, we will get significantly different prognosis. So some evolutionary histories were relatively benign, some were very malign. So this seems to be potentially a very useful way of analyzing cancers. So I've spoken for about 3/4 of an hour, and I want to keep some time for any of you in the audience to ask some questions, because I appreciate it's very difficult to interrupt during the talk, but there still needs to be some interaction, at least at the end. The kind of thing that I was trying to make clear to you is that many people talk about big data and machine learning, but typically in situations where this type of data is unstructured, where we have lots and lots of data in a process that we don't need to interpret, and we can assume the data to be identical from the point of view of its generation. But in reality, in many relevant scenarios, we do have big data, which is made up of lots of slightly different small data, and we must make predictions for the small data. So we must give a prediction that is relevant for that individual or that group of cells or that group of individuals. From a methodological point of view, I'm very fond of Bayesian or probabilistic methods because they have very transparent semantics. And they can quantify uncertainty at each stage of the process. So if I have a model like the one I've shown you from methylation, I can always say, oh, for this group of cells, I'm not particularly certain about this gene here, okay? And that is potentially relevant because then I can go back and say, well, can we measure this gene better, for example? In general, the other thing that I wanted to make clear to you, and perhaps some of you are thinking about in the future now to work in this sector, now biomedicine is a very interesting area. First of all, because we are dealing with some of the more fundamental questions in science, which have a direct relevance for society. But secondly, also because from the point of view of machine learning, this is somewhere where the real questions are. Now, the first wave of AI, essentially, since the big data revolution in the last 10 years, is coming towards the obstacle where we now really need to understand how things work. Okay, we want to use AI in applications where we must be able to understand how certain conclusions are reached and use AI to identify how we could act. And biomedicine gives you lots and lots of relevant, interesting questions where you are forced to develop new machine learning in order for that to happen. And so with that, I will stop my presentation and go back to the StreamYard window where you can post your questions in the chat. So I do have a question here. How are studies on the behavior of cancer cells in space evolving? So how are studies on the behavior of cancer cells in space? Now, I'm not entirely sure whether this refers to metastasis, which is obviously, it's an incredibly important question. And I imagine that that is a slightly separate question from the type of data. In principle, you could take, put together all data from cancer from a patient, so multiple regions within the original tumor and different metastases, and try to work out what is the evolutionary history, which is something that you can do as well. In terms of the mechanisms of how cancer invasion happens and so on, there are lots of both theoretical and empirical works on that topic. I haven't personally worked on it, but I know that it's one of the areas where a lot of models such as cellular automata and stochastic spatial-temporal processes are used and produce very interesting predictions. So far, there isn't an enormous amount of machine learning in that direction. Now, any other questions? I imagine the questions will pop up on the screen. Okay, excellent. So it looks like we got to destination, and as I said, it was great pleasure talking to you guys. And if anyone has additional questions or wants to get in touch with me, you can find me everywhere, at the CISA or in Edinburgh. Thank you very much.